All right, well, hopefully you enjoyed that video and you saw what it was like for him when he can't communicate within his head, right? He had to somehow figure out a way to share the information with himself outside so that then the other part of the brain could hear it, see it, feel it, so that he could proceed on. Now, did you notice in that one section when he was explaining why he chose Bell out of the list of all the other options? That's really common when people are having weird experiences like my corpus callosum is cut and so I can't have thoughts inside my head and now I'm trying to figure out why would my brain come up with something like that. I'm not sure why. It's called confabulation, when you sort of make something up on the fly, right? It sounds logical to him, he said. Right? He goes, I don't know, makes sense to me. But he was just sort of making it up because why would you pick bell when there's trumpet and there's drum and there's like all these other truly musical instruments and you chose bell? Well, he made something up real quick to make it make sense for why he would have behaved that way. So that I thought I'd throw in that separate little concept for you, just as a little bit of fun. All right. So where do we get the information that we have to allow us to understand what a brain is actually doing or what part of the brain is the most important? Now, I've addressed this a little bit in my chapter one lecture, but I want to go a little deeper. I found this picture in an autopsy report of people from Italy who had had blood clots as a result of COVID-19 infection early in the pandemic. The like I like to call it the OG COVID-19 when it was really the really bad version. They were having these clots, and so here's an example of a person who has what we would call a brain lesion now. After they died, they autopsied the person, and you see a lot of missing tissue. Neuropsychology oftentimes will look at lesions and then now knowing what we know about what that region of the brain does, draw inferences about what effects this person would have had if they had lived through it. But even more importantly for psychologists, especially because the report I'm describing came from medical doctors. For psychologists, a lot of times we're on the other side. The person's still alive, but they've had some kind of harm to their brain, a stroke, an injury, an infection, something like that. Actually, the example that I just showed you in that video is an example of neuropsychology where this person has had his corpus callosum cut, and then now they're doing neuropsychological tests on him to determine the extent of effects that surgery has had on his behavior. So for psychology, the biggest impact we tend to have is on that side where we're assessing the behavior. We're assessing, I mean, those experiments that they were putting him through are examples of trying to figure out, can't lead him, can't cue him to what's going on, just try and set, set up the circumstances in such a way that he will reveal the deficits that he has, right? Neuropsychology is very big on trying to figure out ways to get the person to reveal any deficits that they might have without maybe suggesting any deficits to them or causing any changes in behavior that aren't a result of the damage. So it's not just about dissecting the brain and seeing the damage. It's more from our side as psychologists, looking at the behaviors and those changes. All right, so that's neuropsych. Neuroimaging would be the different techniques that we have available for sort of looking inside the brain. And a lot of times we want to just look at the structural factors, right? So maybe we want to administer an MRI, and here we have clearly a tumor growing in this person's brain. So the MRI can give us information about the structures of the brain and if there are any alterations to those. But for us in cognitive psychology, we oftentimes want to know, well, what part of the brain is most important for completing this task or that task? Speaking or reading or solving a problem or thinking of words on the fly or recognizing the color red, or like what parts of the brain are invoked or are most active in that. And so we have fMRIs available to us. And I described this before. This is where they do an MRI, but in addition, they are tracking the blood flow. And the assumption is that the blood flow will be greater in those areas that are the most active, because that's what your brain doesn't have any resources of its own. It doesn't have fat stores or anything like that. It has to rely on the glucose that's in the free floating blood that it's consuming right now. And so the areas that are the most active will be consuming the most blood. And so you do the MRI tracking this blood flow and then let the computer colorize those parts of the brain that are uniquely being activated during this activity, areas that aren't being activated in other activities. So the fMRI is a really strong, powerful tool for us, right? PET scans are very similar to the fMRI, but in the PET scan, they inject a little slightly radioactive glucose. And so in the PET scan, it's actually tracking the glucose and where it's being consumed. And in both the fMRI and the PET scan, the hotter the colors, yellow, orange, red, the more activity, the cooler the colors, the less activity. So we have these neuroimaging techniques. Now, the positive and the negative about these techniques are that, yeah, it's indicating where in the brain we're experiencing activity, but it doesn't tell you exactly when. Is this activity a function of the second you started to think about the problem, or is it downstream from, okay, so you thought of the problem, you started thinking of some solutions, like we don't have a pinpoint of time in the fMRI and the PET scan, but we do have a pinpoint of the location of activity. Electrical recording, on the other hand, which is usually going to take the form of an EEG, where you put a cap that contains electrodes and it tracks the activity of the neurons below those electrodes. The nice thing about an EEG is that it indicates when the activity happens. So it picks up the second there starts to be a burst of activity, it starts picking that up. And so we go, oh, this region started to fire, 500 milliseconds after stimulus was introduced. Okay. The problem with the EEG is that it's not precise in location. It's giving you a region of activity. It's not giving you a precise location of like the nucleus of the source of this activity. So you can kind of see that you've got, okay, well, do I want to know exactly when, or do I want to know exactly where? Because these two different types of techniques are going to give me different kinds of information. We see EEGs used a lot in sleep studies. You might have, if you took Intro to Psych, seen this chart that I have here on the screen, which is showing you the different brainwave patterns that we go through as we go through different stages of sleep. So really commonly in sleep, because we don't care as much exactly where anything exactly is happening in sleep, but we are much more interested in when. How long after onset of sleep do you enter REM? Right? Like that's what we're interested in with sleep studies. They also use EEGs in these ERP studies, like I was mentioning before. We give you some kind of stimulus, and then we track which parts of the brain are activating and how quickly they're activating after that stimulus, right? So maybe I give you, well, I might as well take a research study that I always laugh at on Big Bang Theory, where Amy is supposed to be a neuroscientist, and I think they're calling her a neurobiologist. But anyway, she has this monkey, and she shows the monkey scary things, and then wants to see how his brain reacts to scary stimuli, and how he reacts to happy stimuli and stuff like that. And so she's basically doing ERP research on her monkey. We do this on humans all the time. So we have different ways of getting at the what's going on inside the head, but none of them are perfect. A lot of researchers will try and like combine techniques, so maybe they'll do fMRIs and EEGs in not at the same time, but in sequence. So you get an fMRI, then we do an ERP study on you or something like that. But you can imagine maybe some neuropsychological studies of a person to try and overlay with their fMRI or their EEG results. You can imagine the more things that you combine, the better explanatory power you're going to have. So it's great if you can combine, and that's a great strategy. But we have these things look super scientific because they are producing outcomes that make it seem like you really know what's going on, But the truth is, we have to take these things with a big grain of salt and recognize that we're at the very beginning of using these kinds of techniques. And we may find out that, well, just 2 years ago, they realized that the software that's used to colorize the fMRIs in most research that had been produced to that point had a flaw in the software and it was colorizing things incorrectly. It wasn't grossly incorrectly. It wasn't saying something was active when it was completely not active. It wasn't that degree of wrong, but it was enough wrong that it led to incorrect conclusions. We're right at the baby beginning of a lot of these techniques. Well, everything on the right. And so it's don't put too much, just because there's a picture next to it that looks sciencey doesn't necessarily mean that we can be super sure about the meaning of those results. All right, well, that concludes chapter 2, so I will be seeing you next in chapter 3.